Hello everyone and welcome to another Stat 437 lecture video. In today's video we're going to be talking about what longitudinal data are. So the whole course is about how we analyze different types of longitudinal data, and so today we're going to talk about what are the features of longitudinal data, what do we mean when we're talking about longitudinal data, how do they differ from the types of data that you're likely to have seen before, and then sort of why do we need an entire course dedicated to the issues of analyzing longitudinal data. So with that, we'll pull open these slides over here, and we can ask the question, what are longitudinal data? So to introduce this concept, I'm going to first talk about this study here. So this is a study from the NIH and published in April 2021. And the headline of the study in the press release is that a lack of sleep in middle age may increase the risk of developing dementia. And so my question to sort of get thinking about what longitudinal data are is, what would a study need to look like in order to be able to conclude this conclusion? What would it have to, how would we have had to have worked through this study in order to come to this conclusion? And I ask this not really rhetorically. I would encourage you to pause the video and think about what types of study could have actually led us to concluding this result. So to sort of prompt that thinking, can we conclude this by taking a sample of elderly individuals? So people who are actually at risk of developing dementia. Can we conclude this just by looking at a single sample of the elderly? And the answer is no, because if we've only looked at elderly individuals, how do we know how much they've slept 20 years prior? Now, we could ask them, how much did you sleep when you were in your 50s, say? But the problem we're going to have there is now we're asking someone to remember what was their sleep like 20 years ago. And I don't know about you, but certainly for myself, if someone was asking me how did I sleep when I was, in my early life, I don't really think that I would give a good, actionable answer. I would have to sort of rely on ranges. And when we're talking about something like sleep, people might only sleep a difference of 1 or 2 hours a night, and that might be sort of what's driving this effect. So, no, looking at a sample of only elderly individuals is not going to work. So, can we conclude this by taking a sample of only people who are sort of currently in middle age, right? This way that they'll know exactly how much they're sleeping each night, or we could record it, or we could ask them to keep a sleep journal. And again, the answer is going to be no. And the reason here is because how do we know who's going to develop dementia, right? If all we're recording is how much they sleep during middle age, well, then we don't know sort of the outcome. So can we take samples of middle-aged people and elderly people and somehow combine them? And of course, the answer is no, right? We don't have any way of saying this middle-aged person corresponds to this elderly person if they're samples of different people, right? Again, we would need to know how much were the elderly people sleeping when they were middle-aged, or we would need to know which of these middle-aged individuals is going to go on to develop dementia. And so the only way that we're going to be able to come to this conclusion is if we take a sample of individuals and we follow them from the time that they are middle-aged through to the time that they start developing dementia. And we record how did they sleep when they were middle-aged and who develops dementia. And then we could take a look, after following these people over a long period of time, is there an impact of the amount of sleep you have with your risk of developing dementia? So we have to follow the same set of individuals over a long period of time, and when you're doing that, and you're taking repeated measurements, and you're keeping the same people enrolled in the study, that is what we refer to as a longitudinal study. So, to use a diagram here, we have sort of this representation, the standard representation of a longitudinal study. So you can see that we're dealing with just one group here, group 1 say, and we follow group 1 throughout time. So time point 1, and then we have time point 2, and then we have time point 3, and we're following the same group of individuals over this time. Now this is in contrast to what you would normally think of with a cross-sectional study, which is most of the data you've likely seen up until now. This is cross-sectional in nature, and so instead of following the same group as time goes on, you're going to look at, say, 3 different groups of individuals, right? So this might be young people and middle-aged people and elderly people, but they're taking it at one sort of point in time, and group A is distinct from group B, who's distinct from group C. Now, if you've taken a time series course, you might notice that longitudinal data and time series have, sufficient overlap in terms of their structure, right? And so in fact, a lot of the methods that we use in longitudinal data are related to methods from time series analysis. So if you're familiar with time series, then some of this is going to sound familiar. The key distinction with whether we're doing time series analysis or longitudinal analysis has to do with whether we're looking at a large sample of individuals followed over sort of a few time points, versus in time series, it's more common that you might only have an observation of one individual with very many time points. So for instance, if you're looking at, say, the weather of a single location, right, the weather in Waterloo, you can generate a time series of that because you could take the weather every single day for 25 years, right? So you have one individual, a lot of time. If instead we're looking at sort of the educational trajectories of students in Waterloo, then we have a lot more individuals, and the total time, or the total number of observations of time points that we measure them at, is limited. But again, there's a lot of sort of overlap in all of these types of techniques, so these categories are a little bit fluid. But generally speaking, that's what a longitudinal study is. So formally, a research study in which subjects are followed over time, which typically involves repeatedly measuring the same variables, and they differ from cross-sectional studies and time series studies, as we were just discussing. So you might be thinking, that's nice and all, but why would we care about this, right? Why do we care about these types of studies? Maybe you're not wondering this, but humor me for the sake of this lecture. So there's a handful of uses for longitudinal studies that only longitudinal studies can sort of give us the answers to. So if we want to detect a change in an outcome over time, we need a longitudinal study. Okay? If we want to know at both the population level or the individual level, how does time impact some outcome that we care about, we need longitudinal data to answer that question correctly. So if you want to know, how does your risk of dementia change as you get older, we cannot answer that question without using longitudinal data, because we need to be able to track these individuals and see, as time goes on, what has their outcome done. If we're trying to do this with cross-sectional data, the best that we can sort of hope for is looking at associations between variables. People who are older are more likely to have dementia, but we can't say how does that risk change over time unless we are following the same group over time. This also lets us break down what we term longitudinal effects as compared to cross-sectional effects, right? So longitudinal effects are the effects on an outcome over time. Cross-sectional effects are the effects between two groups of individuals. So the way I would think about this, and the way that we're going to talk about a lot of this over term, is whether we're talking about within subject variation or we're talking about between subject variation, right? So you might find that if you have a group of two separate groups, say, and one of the groups is older individuals, elderly individuals, and one of the groups is younger individuals, right? We can talk about the cross-sectional effect of the difference of propensity to develop dementia, right? The elderly individuals, there's going to be a higher rate of dementia in that group than there will be in the group of young individuals. But This is a different effect. So that's a comparison of two groups. This is a different effect than if we were to follow one individual as they go from being young to being elderly, and we look at how does their risk of dementia change. That would be a longitudinal effect, because we're talking about within a subject over time versus two groups comparing. The other point, and we made this point when we were talking about the dementia study, is that longitudinal studies help us ascertain exposures correctly, right? So in theory, we could have, interviewed a group of elderly individuals, asked them how they slept 20 years ago, and then use that information to perform this. But we wouldn't really be able to know, are they being truthful about their exposure? It might not be that they're lying, they might be misremembering, but we won't necessarily know 20 years ago, how did you sleep? That's not going to be a question that we can reliably answer. If we follow them from the time that they're middle-aged until they're older, we can correctly measure, using journals or actual measurement techniques, for instance, how are they sleeping when they're middle-aged, and therefore we know what was their exposure. Same thing if you're thinking about looking at the effects of smoking, say, right? Trying to ask someone what were their smoking behaviors like 10, 15 years ago is a lot different than being able to measure it as you go on. So longitudinal studies help us in correctly measuring exposures that we might care about. There's the different sources of variation. So this is related to the point that I have above, but you have between and within subject variation. So any individual over time is going to have an outcome that changes. Say we're looking at something like blood pressure, right? As I go throughout my day or over the years, my blood pressure is going to vary within myself. But if you take a population of individuals, you're also going to have variation in blood pressure between people, and even between people who are otherwise quite similar, right? If I look at a cohort of PhD students and myself, we're not all going to have identical blood pressures. And so there are these two sources of variation. There's between individual variation and within individual variation. And in a longitudinal study, we can break down and say how much of this variance that we're observing is within a subject and how much of this variation are between subjects. And so that sort of lets us understand what is the actual mechanism that's occurring here with sort of more detail than we'd be able to if we were only able to detect the between subject variation. We also can detect time effects. So we can look at time effects on their own. How does your risk of dementia increase just as time progresses, right? We know that as you get older, you're more likely to develop dementia. And so that's just a straight up time effect. We can also look at how time effects interact with other relevant factors. So how does prolonged lack of sleep interact with the ongoing time to affect the risk of dementia, right? So we can just use time as a variable in these models and explain, does time have an effect on its own? Does time, when you're looking at time crossed with some other term that we might care about, does that have an effect that's different, right? So we can start to explain things with regards to time, which is useful in a number of scientific disciplines. So the bottom line is that there are many questions that can only be answered using longitudinal data, or which can be best answered using longitudinal data. Even when we can frame a scientific study with regards to cross-sectional data, there are times where the longitudinal data are going to be more effective at answering those questions. And so because of this, because it comes up all over the place that there are questions that we might have that we need longitudinal data to answer, we probably should learn how to analyze this data. And so that's sort of the motivation of why we care about longitudinal data. And so then the next question is, why are they special, right? Why do we need our own methods to deal with longitudinal data, right? You've taken a number of stat courses already. Why is what you've learned not acceptable for longitudinal data? And so what makes them more difficult to analyze? And I'd encourage you to think about this for a second before I actually put up the answer here, right? So what do you think might be the rationale as to why longitudinal data are uniquely difficult to analyze? The answer is that they're correlated. So oftentimes when you get a stat question, we're going to see something like, let x1 through xn be independent and identically distributed random variables. They're IID. And this assumption does not hold when we're talking about longitudinal data. And the reason is that within an individual, any time that you're taking a measurement of the same thing over time, we expect those measurements to be correlated. We expect that, if I have high blood pressure on one day, some component of that is going to be because I have higher than average blood pressure. And the blood pressure the next day, you're going to expect to be high as well. So because these data are correlated, they can't be independent. And because they're not independent, any methods that we're using that assume independence are not going to work correctly with these data. And because of that, almost everything that you've learned is not going to produce valid inference when we're talking about longitudinal data because of this correlation structure. So the obvious question is, how do we deal with that? And that's sort of what we're going to be doing with this course. Welcome to Stat 437. We're going to spend the rest of the time learning how we deal with the fact that we have these data that we want to answer important scientific questions about or with, but that we don't know how to analyze properly because they have correlation within them. Okay? And so from here, I just want to run through a few example data sets and hopefully reinforce some of what it is that I'm talking about here. So these data sets should all be posted in one form or another on Learn, and I'd encourage you to play around with them. And hopefully there's some more data sets on Learn other than what I have directly here, but briefly, let's run through them. So the first is the TLC trial. So it's the treatment of lead in children, and it's essentially a trial that's looking at the quantity of lead that is observed in children's blood. And this comes because children, I think for the most part, children eat paint. They lick walls, paint flecks come off, they eat those. And so if you have lead paint, children get lead in their bloods. And so this was a trial that was run to compare two different treatments, a placebo and an active treatment. And so we can see here, we're looking at this data frame. Just to walk you through it quickly, this first column is just the ID, so there are 100 children in the study. This column here, this treatment column, says whether children were on a placebo, denoted with a P, or an active treatment, denoted with an A. So this was the experimental treatment that they actually had. And then over here we have 4 different measurements for each child of the level of lead in their blood. So it's at week 0, so the baseline of the study, week 1, week 4, and week 6. And so we can sort of ask a number of important scientific questions about this. So the first and obvious question from the trial is there a difference between the placebo and the active treatment, right? We also might just want to know, over time, how do children's blood levels change? And we could want to know this overall, right? Does the level of lead in their blood decrease over time if they're not continually exposed to more of it? Does it increase? What's happening there? But we might also want to know, if they're being treated, how does this change over time versus if they're in the placebo group? How does this change over time? And we could also ask, assuming that there is a change over time, is that change over time the same whether they're in the placebo group or in the active group? So when we're talking about a medication that's supposed to have a longitudinal effect, so it's supposed to, say, decrease the amount of lead in blood over time, then we would want to use a longitudinal study, and we'd want to ask about this time effect of the treatment. And so these data are fairly straightforward, but they should be up there, and encourage you to take a look at them. There's also some sales data here. And so I believe that this is sales data collected from grocery stores about pasta brands. And so you can see here we have this brand category, and the brands there's 4 different brands. I'm not sure which brands they correspond to. But each of these brands has potentially several different products. And so you can see that sort of each brand product combination is a unique entry. And we can see that these are grouped by calendar date. And so there's just over 4 years of data in this study, or roughly 4 years of data. And the outcome that we're measuring here is this quantity, which is the number that were sold on that day for that brand with that product. And this last column here is an indicator, this promo, as to whether or not that product was marked on sale on that day. And so you can see that sort of we have that same format. We have these different products from these different brands, and over time, where time here is actually being measured not in sort of weeks as the last data were, but in terms of calendar date. And we could say, are the different brands comparable? Are the different products comparable in terms of the amount they sell? The question, I'm sure, from the point of view of the corporations themselves are, how does the promotion impact the amount of the quantity of sales? But also, do the effects over time change? Do they change within the brand? Do they change whether they're on promotion? Is there seasonal effects in this data? That type of thing. And so all of these questions, again, we can only answer because we have these longitudinal data. This next example here is slightly different, and what we have here are a number of different podcasts. So we can see that the different titles for what these shows are. And these podcasts every day were for, I believe, a month, roughly a month. The number of reviews that they had on their iTunes listing, as well as the star rating value, was recorded. And so we have several different observations for each podcast as time goes on as to the number of reviews and the star value rating. And then in this data frame, and if you download the data and open it up yourselves, there's a whole load of other features with this data. So there are features related to the category of the podcast or the last upload date. There's also features related to sort of sentiment scores or mood scores about the description of the podcast or about the reviews that users are leaving. And so here, I think that the obvious questions would be ones of prediction. So can we predict how the number of ratings will change over time, or can we predict how the average rating that a podcast receives will change over time? So again, here we're sort of outside of this medical domain, we're outside of this business domain, but we can see that there's still this way that we can map this longitudinal structure onto these data. The last example that I have here is a slightly different framing entirely, and it's going to be the focus of the second half of the course, but it's longitudinal data in its own regard. And so this is from a stroke trial, and we can see that we sort of have 4 different columns here, and these 4 different columns are slightly different than the way that we had otherwise. So here, time is progressing in years, so there's 10 different years, and then each of these columns represents sort of a different group of individuals. And so there's this 0, 0 group, there's this 0, 1 group, 1, 0, and 1, 1, okay? And the groups are about whether or not the individuals are in a treatment, an active treatment. So this first digit is a 0 if they're in the placebo treatment, and it's a 1 if they are in the active treatment. And this second digit here is about whether they've had a previous stroke or not. So a 0 means no previous stroke, a 1 means that they've had a previous stroke. So the 4 different groups are placebo treatment, no previous strokes, placebo treatment with a previous stroke, active treatment with no previous strokes, active treatment with previous strokes. And then for each of these categories, there's a certain numerator and a denominator. And the numerator is the proportion or the number of individuals from that group that had a stroke in the current time period. And the denominator is the number of people who were in that group at that time period. Okay? And so in this first year, we can see that 57 of the 344 individuals from the placebo with no previous strokes had a stroke this year. And same thing across the rows and columns. And we can go down. And so this data, really, what we're looking at here is time to event data. So the outcome that we'd be interested in is how long until we observe someone having a stroke in each one of these groups. We might also be interested in questions like how probable is it that someone from one of these groups, lasts beyond year 8 before they've had a stroke, and how do these probabilities or these average median time to the onset of a stroke, how do these differ between whether that you're in the treatment group, whether you've previously received or previously had a stroke, that type of thing. So here we have survival data rather than sort of strict longitudinal data that we've been talking about previously, but it's still longitudinal data because we're observing the same sets of individuals over time, taking repeated measurements, and we have to find some way of dealing with the fact that these data are correlated. So like I said, that's just a handful of different examples and some questions that might go along with them. I'd encourage you to think about some different questions that these data might answer. The other data sets that are posted on the course website, I encourage you to take a look there and see if you could come up with some good questions for those, or data that are outside of this entirely. I'd also encourage you to sort of discuss this with myself. Send me an email, send me a message on the course form, or with your peers as well. Longitudinal data are sort of hiding everywhere, and if you have a particular interest, if you have a hobby or something that you're particularly into, there's a good chance that there's some way that we can apply longitudinal data to that subject. And if we can find that and it seems interesting, then I think that's a great way of trying to engage with the course material. And so I've tried to give you a wide range here. Oftentimes you're going to see these used as medical or biostatistics type studies, but they don't have to be, right? We looked at podcast data, we looked at sales data. You could take a look at sports data, for instance. It's all going to sort of fall into the same bucket, and our methods are going to be useful across the board. So certainly if you have a thought about something, let me know. Be glad to discuss that with you. But for now, we can jump in and just summarize what it is that we talked about today. So longitudinal data are data that occur when we take repeated measurements of the same individuals over time. Oftentimes we're measuring the same variables over and over again. Longitudinal data are required for answering questions about changes within an individual as compared to changes between individuals, and to capture time effects. So there's no way of answering these types of questions without longitudinal data. And longitudinal data are challenging to work with because the data are correlated. So the reason that we need specific techniques for handling longitudinal data are because these data are correlated. If they weren't correlated, we wouldn't need these specific techniques. So with that essentially wraps it up for today. I hope that gave you a good sense of the overview of what longitudinal data are and gives you a sense of the types of questions that we're going to try to answer this term. And I will see you in the next video.